So she may not have won the Nobel Prize, at least this year, but Katalin Karikó is this amazing scientist who is responsible for the mRNA vaccines that many of us, including myself, are benefiting from. He made this longer post, and so I wanted to walk you through some of the basics. And so what I'm going to focus on is like the scientific discoveries she made, and kind of take you through some of the papers and some of the key figures about how she was able to figure out how mRNA could get used for like protein replacement and for vaccine. Karikó is a pretty fascinating story. So she believed in the potential for this mRNA technology to help people when like no one else believed in her. She was actually demoted by the University of Pennsylvania, UPenn, in 1995 because she couldn't attract funding. So they said that she would either have to switch to a different project or accept a demotion if she wanted to keep working on this mRNA stuff, and she wanted to keep working on this mRNA stuff. And so I am so glad that she kept doing it, and so mad at UPenn that they didn't. But anyway, so then she published this groundbreaking work I want to tell you about in 2005, but it didn't get very much traction. Then in 2013, UPenn refused to undemote her to a tenure track position, so she left and joined BioNTech, which as senior VP. And so BioNTech is the one that's working with Pfizer to make a lot of the vaccine that we depend upon, including the shot that I got. And then in 2020, as of March, she was cited about 1,500 times, and as of March 2021, she had been cited 600 times already, and I haven't updated this since I originally made this figure then. But it's pretty amazing. And so she's been super underappreciated, and I want to tell you more about her and her work that she did. So the basic idea is that you have just a little like reviewer of the whole like central dogma of biology is that you have a gene that's in the instructions for like making a protein written in DNA. Then that gets transcribed into messenger RNA copies. That then that process is called transcription, where you're going from the DNA gene to the messenger RNA, and then that gets made into protein in a process called translation. So you have gene, transcription, mRNA, translation, protein. So basically, the whole purpose of kind of like transcription is just to make lots of messenger copies of the RNA. And normally in your cells, that's happening like in the nucleus. So it's like a membrane bound room inside of your cells. So you can make lots of copies of this messenger RNA from the DNA. And that way you don't have to keep going back to the DNA, which is risky because that DNA is like your permanent like instruction manual. And also, that's happening in the nucleus, and in the cytoplasm is where the cells are. Where the ribosomes are, which are the protein-making complexes. So you need to get those messenger RNAs where the protein makers are, so they get exported into the cytoplasm. So that's what happens in your cells. But it turns out that you can just give, instead of doing all that transcription like in your nucleus, you can actually just give cells messenger RNA into that cytoplasm, and that's where the ribosomes are all ready to go, and they can use that those mRNAs to make protein. And so this was the idea that Karikó had. Well, other people had this idea too, but no one was really having that much luck with it because they were running into a bunch of problems that I'll tell you how she figured out how to get around. But basically, she figured out the promise with this technology was that if you could make and deliver this mRNA and get cells to make it, you could do things like protein replacement. So if there was a disease that was caused by like a missing or damaged protein, you could give the instructions for making that protein, and then the person's own cells could make it for them. Or you could also use it for vaccines. So get the cells to make a foreign protein, like the coronavirus spike protein. And then the body can learn to recognize it, make antibodies against it, etc., without you actually having to get sick. And so this is what is the basis for the mRNA vaccines against COVID-19, where so like in the injection you have these mRNAs for the spike protein encoded in these lipid nanoparticles, which kind of help it slip into cells. And so I'm not going to talk much about that, but I want to tell you more about this technology that makes it possible. So when Karikó was getting started, some people had showed like basic proof of principle stuff that basically it was possible. And so I have a lot more detail about all of this in this article this blog post that I wrote, as well as a bunch of sources and this papers that I'll be talking about. But people had shown that it was like possible, but there were problems. And so basically, just like a quick reminder, so DNA and RNA are basically like chemical cousins, kind of. So they both have these, they're made up of nucleotide letters. So ribonucleotides for RNA, deoxyribonucleotides for DNA. So they don't have that OH there, but they have these groups and they can link up. And so they have these base parts, which are like these unique parts. So it's ACTG in DNA and ACGU in RNA, but you can pair with A. So you have these specific base pairing, and this allows you to use one strand as a template for making a second strand and that sort of thing. So just to get a little familiar with some of the terms that I'll be using, so you have the base, which is like, so you have this sugar part, then the base is the like unique part, so this AG UC part, and then these like phosphate groups, which are what gives it like the energy to link together. And typically these are put together by enzymes, so like protein helpers called polymerases. Okay, so that is the background for what you'll need. Okay, so they found that one of the reasons why they were having difficulty putting this technology to use was that when they went to put in the RNA, it was causing the body to, like, well, they weren't doing this in humans, but if they did it in like cells or in laboratory animals, it was causing like this immune reaction. And so when they put in the RNAs, like no matter what RNA they put in, like no matter what the sequence was or whatever, it would just like the immune system would kind of like get set up. And so here's this experiment where they're showing this, and so here the alarm that they're measuring is levels of TNF alpha, which is a type of chemical messenger called a cytokine. And so basically it's this thing that gets released by like in response to when there is a like the immune system gets set off, basically, and so it can then in the body and stuff trigger other pathways and that sort of thing. But here they're just using it to show that the immune system is getting activated. And the part of the immune system that we're talking about here is part of the innate immune system. So that's not responding to something specific, like it's not specified to like specifically react to the protein of the coronavirus or that sort of thing. It's like any foreign RNA. But how does your body know that RNA is foreign? Because your cells are filled with it, right? Your cells are filled with a lot of RNA, and so it needs to be able to know what is foreign, and what is not foreign. So they took RNA that they had in vitro transcribed. And so remember, transcription is going from the DNA to the RNA, and they've done this in vitro, so like in a test tube. So they made it like that, and when they put it in to the cells, you can see that they get this higher immune response. They also get a really higher immune response when they put in most like the total E. coli RNA. But something interesting was that if they put in bacterial tRNA, which is transfer RNAs, which is a type of helper RNA that's used in the translation process, it like hooks up to the amino acid to be added, so that's like a protein letter, and it forms as the link between the mRNA and the protein. So basically on one end it has this little like 3 RNA letter anticodon that matches to a codon, which is like a word in the mRNA, and it brings the corresponding amino acid, and that's how you the letters get put together and that sort of thing. But anyway, the point here was that the bacterial tRNA was much less immunogenic. So if you see the total RNA versus just the tRNA. For mammalian RNA, the total RNA is much less immunogenic. However, the mitochondrial RNA is much more immunogenic. Okay, so the stuff they made in vitro was really immunogenic. Bacterial RNA was really immunogenic, whereas mammalian RNA wasn't. But the exceptions were mammalian mitochondrial RNA, which was, and bacterial tRNA, which wasn't. The plus minus refers to with or without an RNase, which is an RNA degrader that they added to show that the responses were specific to RNA. So even in these cases where you have like a high immunogenicity, so this high response, if you add the RNAs, you don't see the response. So the reason why with like this R848 or whatever, like this is a different compound that is just like a positive control to show that the response, like it activates the immune system, but it's not like an RNA. So that's like why you still have it with the plus. But it's like a compound that'll do that. But these other ones rely on the RNA itself. Okay, so what do these RNAs have in common? Well, it turns out that mammalian RNA, so the mitochondria, so you might have heard of like this endosymbiosis theory or whatever, where so the mitochondria are kind of like the powerhouses of your cells. And they are the, like where the energy making takes place. And they're actually like this membrane-bound compartment. And it is thought that it arose when like a eukaryotic, so like, eukaryotic is basically mostly like anything other than bacteria. It kind of like, it has membrane-bound compartments inside of it. And so it's like plants, animals, all of that stuff. Yeast, all of that. And so it's thought that the mitochondria actually arose when like a early eukaryotic cell like swallowed a bacteria. And then so the bacteria have their own DNA and everything, and so we actually have some mitochondrial DNA that's different than, like, that's extra, and it makes some mitochondrial RNA. And so this mitochondrial RNA, because it has this like bacterial origin, is more bacteria-like. And so there's something that the bacteria and the mitochondrial DNA or RNA have in common. And it turns out that what they have in common is they're highly modified. They're not highly modified. So the tRNA, the bacteria though, is, and all of this other RNA in our cells is highly modified, whereas the mitochondrial DNA, RNA isn't. But it's not really a problem because it's in our cells. I mean, it's like bound up in a membrane compartment, so it's okay in there. But there's something about unmodified RNA being out in the cell that was causing this signal. So that could sort of serve as a trigger, like, oh, this should not be here. So it's okay if the modified RNA is here, but not if the unmodified RNA is here. So that's a nice theory, right? But how could they test it? So here is just showing that again. So here's the percent of the modification and the activation. So you can see that total mammalian RNA is very low, but they have a high, and the mammalian tRNA and the E. coli tRNA, they're all very low in terms of activation, and they have a high level of nucleoside modifications, up to like a quarter. Whereas the mitochondrial total RNA and the E. coli total RNA, they're barely modified and they give a high response. So this is just shown in a table, and this sort of thing, and links to the articles that mention them. And so I won't go further into that, but right now this is just a theory, right? So we have a correlation, but we haven't proved causation. So how could they show that it was the modifications that mattered? So they made, basically, going back to in vitro transcription, where you can make the RNAs in like a test tube, basically. And so they made synthetic mRNAs with various modified nucleotides. And so nucleotides, remember, like the RNA letters. So they made specific modifications to these, the base part, so this like part that sticks out. And so and then tested their immunogenicity. So how much of that TNF alpha signal did they produce? So they found, so here's just to orient you with the scale. So this is showing like the amount of modifications. And so there's 1571, basically this RNA is 1,571 letters long. And then the y-axis is showing the incorporation. So least modified to most modified. And so remember, the higher the signal, so this is showing inhibition of the TNF alpha expression. So the higher the signal, the less the immune response. So it's kind of like opposite of what we were showing before. So if it's at 100, that means that it inhibited the response completely. Whereas if it's at 0, this is there's no inhibition of the response. So you get the full response as you do with like normalized to the normal well, I guess that is kind of your normalization because that's unmodified. So they did this with a couple of different modifications. So like methyl 6, methyl 5, C, methyl 6,8, and then the pseudouridine. And they found the pseudouridine was especially effective. And so what's pseudouridine? It's basically this U part, so this base. It's basically just like flipped. It's like rotated kind of. And that makes it so that it can that makes it slightly different, and it's different enough that your body can say, okay, it's been modified, it's all cool, we're cool. And that's that. And so you can see that the more of this that you incorporate, so even with just like at this level, you're getting almost complete inhibition, and so it's more effective than the other ones. But why was it immunogenic? So it makes sense, like evolutionarily, like it makes sense in theory that if you have a modified RNA, your cells would be like, okay, well, we modify our own RNA. Bacteria don't modify their RNA much, so it's probably not bacterial RNA, it's probably our own RNA. But why was it so? So they wanted to figure out what it was about this RNA that actually, like, how was it being distinguished by the cells? And they showed that, at least in part, it was acting through these things called toll-like receptors, or TLRs, which are kind of like a class of protein watchdogs. And so they bind to various PAMPs. So PAMP is a Pathogen Associated Molecular Pattern. So basically, it's like some molecule that's normally associated with a microbe, so your body wants to keep an eye out for them. So just kind of like we have specific things that our immune system keeps an eye out for, that our adaptive immune system keeps an eye out for, like how we have antibodies that can be specifically watching for a specific viral protein. We also have these receptors that look out for kind of more generic things. So these innate immune system, there's things that like most bacteria or many viruses have in common. And so these we refer to these as PAMPs. And so they're molecules that normally, so the cell knows, like, if we see this, it probably means that there's a virus or a bacteria, but it's not like it's just this specific virus that we've seen before. It's not that type of thing. It's not that adaptive immune response. This is innate immune response. So they took cells that didn't express the TLRs. So those cells don't react when they have any of the in vitro transcribed. So that's in this lane. So this is like their control, where this is like they don't have any of the TLRs. And then what they did was they put in DNA into those cells to get them to make the TLRs one at a time. So they took like plasmids, so like a circular piece of DNA that has the instructions for making these TLRs, and they got the cells to like overexpress them, so to make a bunch of these receptors that they wouldn't normally make. So now when they added the, so if they add the unmodified RNA, they get this high response, especially with TLR 7 and 8. But when they add the modified RNA, especially this, so this is the pseudouridine that's highlighted in green, they see a much lower response. And so here the signal that they're looking at is IL-8, which is a different like chemical signal that the immune system gives off. Another chemokine that kind of like helps recruit in other factors. And so they see this difference when they have the modified. It's not setting off these, it's not interacting with and setting off these TLRs, and so you don't get that signal. So they didn't stop there. They wanted to see, okay, well, how is this actually, does this affect the protein synthesis? So you would think that it's great that if you want to make a cell make a bunch of proteins, so such as like for protein replacement. So remember we had those like couple of different things that you could do, like protein replacement, where you're basically like introducing the instructions for making a protein that a person's cells have problems making themselves. Or you also, then we talked about the vaccines. So in the vaccines, you would think, okay, well, you want your body to recognize it as foreign, right? So, but why? So wouldn't you want? So it makes sense that if you have, like, you're doing it for a protein replacement purpose, you would want to introduce those modifications so that your body doesn't mount an immune response. But what confused me at first was like, well, why would you want to do that if you're doing a vaccine? Like, don't you want your immune response? But it turns out that that immune response actually stops the production of protein from that RNA. So in addition to like those toll-like receptors, the unmodified RNAs interact with a different immune sensor called PKR. And what the unmodified RNA does is when it binds to PKR, it activates PKR. And PKR is a kinase, so it's a protein like enzyme that'll add phosphate groups, so these negatively charged groups to proteins. And so when PKR is activated, it actually like autophosphorylates, so it phosphorylates itself when it's bound to the thing that it senses. And what this does is then it phosphorylates EIF2 alpha. So EIF2 alpha is a translation initiation factor. And so you might remember translation is the process of making a protein from the mRNA. And so when EIF2 alpha is phosphorylated, translation is halted. So you have unmodified RNA, you shut off translation, so you don't make the protein. With modified RNA, however, now it doesn't bind, it doesn't activate PKR, and translation proceeds normally. So here they showed that the unmodified RNAs leading to PKR phosphorylation. So here they're showing that with the RNA modification, they're detecting this PKR phosphorylated version, but not with the pseudouridine. And the same with the EIF2 alpha. So remember the so this is showing the self-activation of the PKR and then the activation or the inhibition of EIF2 alpha. And so you get low levels of translation. So with the wild type, and then you get like a 5-fold increase in translation when you have the pseudouridines. Oh, and I didn't mention it before, but the pseudouridines, they still like act like normal uridines in terms of like pairing and interacting with all of everything, and so that's all cool without. So it doesn't affect like the functioning of the RNA; it just makes it less immunogenic. So, and this, if they have cells without PKR, they don't see any difference in translation between the modified and the unmodified. So it looks like if you put in the pseudouridine, you get more translation and you don't set off that generic immune response. But since your body is still making the since your body is still making the like viral protein or whatever in the terms of a vaccine, then your body can still learn to recognize that as foreign. And it can also kind of just like get this generic effect from having the like the just the like stuff that the mRNA is coming in, like the form that it comes in or whatever. It can still generate a bit of an immune response, that innate immune response trigger it, but not like in a way that's going to be detrimental to the actual production of the protein. And it just kind of lets your body know that it is something foreign to keep an eye out for. And so here's the success story. So from all those doubters and everything, and now we have these amazing vaccines. So I had actually made this post back in March when these vaccines were a lot newer, and so now we have even more evidence that they're really safe and effective. And so here's just like a couple of the original papers, so you can see that Karikó is a co-author. And in the method section, you can see that they're using that modified RNA. So here they're actually using methyl pseudouridine instead of uridine, and so it just has this extra methyl group as well. And then, yeah, here's this amazing paper showing that the vaccine actually works. This is showing the cumulative case incidence following first injection. And so links to all these papers in the notes. But yeah, so it's this amazing success story. But Karikó is not done. She thinks there's still a lot of potential for this technology, both in terms of vaccines and as well as the like protein replacement strategies. So some of the pluses and minuses about synthetic mRNA. So one of the great things about it is that it can be easily adapted to get cells to make any protein or a version of a protein that you want, as long as you know the mRNA sequence that you need. So this is what allowed the vaccines to be made so rapidly when they just knew the sequence of like the spike protein. Since the protein is being made in your cells, it gets modified and folded like it normally would. So a virus infects your cells and it gets your cells to make all the viral proteins. And so the viral proteins are made in your cells when the virus infects you. So if you get the cells to make the protein without the virus infecting you, the protein is going to be made just as it would be if the virus infected you. So this includes like all the modifications, like the glycosylations, which is the big one for the spike protein. So it's like it gets a bunch of sugar chains added. And so some of like the vaccines and stuff, like our protein subunit vaccines, like the Novavax. So they actually make the proteins in like insect cells, and then purify those proteins and put it into like the shot. But to do that, you have to make a bunch of the protein in these insect cells, and then you have to purify it, and they might have different modifications because they're being made in insect cells versus human cells, and so that could potentially create a slightly different immune response or that sort of thing. But since the cells are making it yourself, you get past all of those problems. It does not incorporate with your DNA, so there's no risk of it causing mutations to your DNA. It doesn't even get into your nucleus, which is where your DNA is. And the effects are temporary, which can be good for some purposes, like a vaccine. So you wouldn't want your cells to like always be making. You wouldn't want like all your cells to be making this all the time. You just want it made for a short time, and then the mRNA gets degraded and all's good. But there are some challenges. So there's the generic immune responses causing inflammation, which we saw can be greatly improved by incorporating modified nucleotides. However, you still have generic immune responses that like you want, but for a vaccine, but you wouldn't want that for like a protein replacement strategy. So remember, that was like a big hurdle for it in protein replacement, is like getting it not to just set off an immune response when you just want the cells to make the protein and not like learn to react against it. So our bodies can learn to react against it, like the viral proteins, it hasn't seen them before. So it'll be able to recognize them as foreign. But with protein replacement, you're usually introducing a protein that your body has seen before, it just can't make enough of, or that sort of thing. So you don't worry about that, but you still don't want that generic immune response to occur. A challenge is also getting into cells without degradation. So this is where those like lipid nanoparticles come in. So you have to get this like lipidy coat surrounding the RNA, and it has these special properties like that, so that when they're like in cells in the endosome or whatever, the pH change can cause the lipids to become charged, and then so that lipid nanoparticle can kind of dissolve or like come apart, and then the RNA can get out. But it's a challenge. One of the main challenges is like getting it into specific cell types. So this is mainly an issue for protein replacement of non-secreted proteins. So if the protein doesn't get secreted, and you only get it into like cells in your arm muscle or whatever, then you could have, that's not going to help you if the problem's in your eye. It's only going to help in the cells you got it into. And so some of the techniques that are being introduced are things like trying to put things on the surface of the lipids so that they bind to specific receptors and say like the liver, and then get taken in by specific cell types. So that's a very active area of research, is trying to get better targeting to specific cell types. Also for protein replacement, what can be bad is that the effects are temporary, which was a plus that we saw with the vaccine, was that they're temporary. However, if this is a protein that that a person's going to need like re-administered over their lifetime, that can get expensive and that sort of thing. But there's still like a lot of potential, and Karikó is really the like spearhead of all of this, and it couldn't be done without her. So she has this amazing story, and she also worked with, a lot of this was done with an immunologist named Drew Weissman, UPenn, who believed in her when other people didn't. He is an immunologist, and he was interested in the vaccine technology, whereas in the beginning, at least, Karikó, and she still is interested, as far as I could read up and stuff, in like the protein replacement stuff. But he was an immunologist. He was looking into the vaccine aspects, and so they initially worked on like making an HIV vaccine, so they paired up and they did all this stuff, and then UPenn still wouldn't promote her to tenure track position, but she has like this amazing story, and I want you to check this out. And yeah, I'm trying to see if there's anything else. So I think that's mostly it. So one more thing I wanted to say was, yes, so although I said like you just need to know like the sequence, it's not quite that simple. So another challenge is trying to like get to optimize the front and end matter kind of the mRNA. So like the cap and the tail, like in the UTRs, so the untranslated regions, to try to like maximize the expression without the actual sequence part. And then there can also be like different splice variants, so basically different ways that the gene has introns and exons. So I don't think I have the slide here, but the introns are like regulatory regions that can you have that then get like spliced out, and they can do this in different ways to give you different splice variants. And so sometimes a different one splice variant might give you better production than the other or whatever. And you can also optimize like the lipid nanoparticles. So there's a bunch of stuff that you can optimize, and there's a bunch of so it's not quite as easy as just like knowing the sequence. But yeah. Oh, and one more thing. So the reason why some of the early experiments were failing or whatever, is that your cells also have receptors for recognizing double-stranded RNA as foreign. So we talked about how they're able to recognize unmodified RNA as foreign. They're also able to recognize double-stranded RNA as foreign. Um, and so some of the early experiments were failing because they were introducing a response because the in vitro transcribed mRNA. So even when it was like, sometimes even when you had the incorporation of the modified, you'd still get some response and whatever. So they found that there were actually some like double-stranded RNA contaminants from the in vitro transcription process, and so by better purifying the components, so better purifying the mRNA, you could get better expression and don't have that response.